everyone. I hope I'm audible to all of you. So today, this is the short video about the solutions of gatexe Microbiology 2023. Okay, so in this video, we will see and we will discuss the solutions for the question paper which was there for the gatexe, that is Life Sciences exam in 2023. So the first question was, monkeypox is caused by. So, monkeypox, it is basically caused by a virus. Okay, this is the disease that is caused by the virus. And the thing is, this is the member of orthopox virus. Okay, and this is of the genus. This is of the family Poxyvirus. So here the question is whether the monkeypox it is caused by the virus that we know, whether the virus is going to be double-stranded DNA, single-stranded DNA, double-stranded RNA, or single-stranded RNA. So as we all have studied that the virus can have genetic material as DNA or as the RNA. So this virus, it is basically the oval-shaped virus it is oval-shaped and it has double-stranded DNA genome. So your answer will be option A, that is double-stranded DNA virus. Now the second question is, which of the following is going to convert the sulfate to hydrogen sulfide? So the options are your D-sulfovibrio, Beggiota, Thiobacillus, and Thiotex. So for this solution, we have to go for the go for your sulfur cycle, like how it is going to happen. So here in this figure, you can see there are basically three steps. The first one is your assimilative sulfur reduction, then mineralization is there, and the sulfur oxidation is there. So the question was how your sulfate is reduced to your hydrogen sulfate. Sulfate is your nothing but SO42 minus. It should be reduced to your H2S, that is hydrogen sulfate. So here in this cycle, you can see this SO4 is reduced, like it is converted to H2S, or it is being reduced to H2S, or it is converted with the help of which bacteria? So the bacteria is D-sulfovibrio. This is known as sulfur reduction. Okay, this is the process of reduction that is known as the the bacteria which is there for this process is known as D-sulfovibrio. And we all know what is oxidation and what is reduction. So addition of hydrogen is your oxidation, and your release of hydrogen is nothing but your reduction. And also you can see if the question was like sulfur oxidation, how does it takes place? So that was basically your S naught is converted into SO2 3 minus, SO3 2 minus, and the bacteria responsible for sulfur oxidation are here you can see. And when you are SO3 minus, this was your sulfate, SO4 2 minus. When it would be SO3 2 minus, then for reduction of that, you will require the bacteria, not the desulfovibrio is there, another one is Clostridium and Ultramonas. So all these bacteria are there for a specific step. Suppose it was sulfur reduction, then the bacterial name can be changed from S naught to H2S, Desulfromonas, cyanobacteria, thermophilic, and archaeobacteria. And thiobacillus would be there, which are aerobic. That would be from H2S to S naught. Okay, so according to the step that is going to happen in sulfur cycle, your bacterial species will vary. So for this question, the correct answer is D-sulfovibrio, that is option B. Now moving to third question. That is your which one of the statement about the bacterial flagella is correct. We all know what is the function of flagella. If this is your bacteria, then here this is your flagella. Basically, the locomotion is the function of the flagella. Now the question is, which one of the statement is correct? Like from four of the statement, which one should be correct? The first is flagella varies in length ranging from 0.5 to 2 micrometer. Flagella are adjacent fibrils with regular pattern. Flagella helps in conjugation. Flagella originates from the basal bone. So, see, the length ranging from that to that micrometer, it is not fixed and it is almost not that short. Okay. The second is the flagella are adjacent fibrils with regular pattern. No. Flagella helps in conjugation? No. What helps in conjugation is your pili or pili protein. And then flagella originates from basal body? Yes, this is correct. It is going to originate from the basal body because the cilia and flagella, that is going to originate from the basal body. Okay. Another name for basal body is the basal granule, or it can be your kinetosome. And the basal body is nothing but the protein structure found at the base of eukaryotic flagella. That is known as the basal body. On that base, your flagella would be, oh, it will originate. So for this question, the correct answer is option D. Now moving towards the next question, that is the correct sequence of metabolic intermediate in Krebs cycle. We all know Krebs cycle is also known as TCA cycle or citric acid cycle. Okay, tricarboxylic acid cycle, citric acid cycle, or it is also known as the Krebs cycle. Krebs cycle, why it is known as Krebs? Because it was found by the scientist known as the Krebs, hence the name Krebs is given. Now in this cycle, there are multiple steps. So here in this figure, you can see basically there are 8 to 9 steps in general. The first is like formation of acetyl CoA from the pyruvate that we have studied in glycolysis. Pyruvate is from, and then from the fats of pyruvate we get acetyl coenzyme A. Now that acetyl coenzyme A, what will happen? It will combine with your oxaloacetate. Okay, that is the last product, and it is going to form your citric acid. Citric. This is your citrate. From citrate, it will form into isocitric acid. From isocitric acid, we get 5 carbon compound, that is alpha ketoglutarate, that is 5 carbon compound. Then from this, further succinyl CoA is formed. Then from succinyl CoA, we get succinate, then we get fumarate. All these are 4 carbon compound. Then we get malate, we get oxaloacetate. At oxaloacetate, your TCA cycle will end. Now see, the question is, the correct sequence of metabolic intermediate in Krebs cycle. So in Krebs cycle, see, here the sequence is given. The first is alpha ketoglutarate, fumarate, succinate, malate. But here you can see after alpha ketoglutarate, there is no fumarate. There is succinyl CoA or succinate. So this option is wrong. The next is from fumarate to malate, succinate, alpha ketoglutarate. This is the reverse sequence. This is not the proper sequence. Okay, alpha ketoglutarate to succinate to fumarate to malate. Okay, this can be considered. Let's check the option D. Succinate to alpha ketoglutarate, malate, fumarate. No, because after alpha ketoglutarate there is succinate. Here you can see alpha ketoglutarate, succinate, fumarate, malate, oxaloacetate, and here it is given this end. As option C is the correct answer for this question. Going towards the next question. The question is, catabolite repression in bacteria is regulated by concentration of, from what concentration your catabolite repression in bacteria is regulated. So basically what happens in the case of bacteria, your glucose and related sugars, they repress the transcription of genes. Transcription is nothing but the formation from DNA. We get RNA or mRNA, messenger RNA. That is your transcription. So the glucose and related sugar, they repress the transcription of the gene and encoding enzyme required for utilization of alternative carbon source. Is encoding enzymes required for utilization of carbon source. Okay, this is the first thing. And now some of this sugar, these are also expressed by the other sugar. So other sugars like galactose, then and this whole process of repression is nothing but your catabolite repression. Now, catabolite repression in bacteria, it is regulated by concentration of what? So from this, what we get is it is regulated by concentration of glucose or related sugar. So here the glucose option is given. So it would be glucose. If glucose is not there, it would be galactose. So correct option is option B. The next question is phagocytosis. Phagocytosis was described by. This question is from our first chapter, that is history of microbiology. So we know phagocytosis was first described by the Elie Metchnikoff, and Robert Hooke here discovered the cell basically from the bark of a tree. Then Robert Koch has many inventory. Paul Ehrlich, it is there for antimicrobial chemotherapy. For that, for drug production, for antibiotics, Paul Ehrlich has a good contribution. So when we go for the phagocytosis, so it was all first seen by the Elie, and it was first reported by the Canadian physician William. But here, who had discovered? That was the question. So correct option is option A. So option A is your correct answer. Next question is, which one of the following statement about batch culture of microbes is not correct? Not correct. Okay. You have to choose the wrong one. The first is cells from the stationary phase will show longer lag phase when inoculated into the fresh growth medium compared to those collected from exponential phase. See, for this, we have to know, we should know the microbial growth curve. You all know, like from here, this is your lag phase, log phase, stationary, and then you have exponential. This is lag phase, this part is your lag phase. This exponential is log, this is stationary, this is decline. The question is, cell from the stationary phase, this is the session, will show longer lag phase when inoculated in fresh growth medium compared to those from the exponential phase. Okay, this is true. See, this is more, na? This is less. This length is less. This is more. So obviously, statement 1 is true. Next statement is, death phase of culture is often exponential in nature. This is the decline phase. This can be exponential in the sense your decrease in number of bacteria would be in the, that would be 90%. Okay. Or one log cycle decrease. One log cycle decrease would be there. Hence, that is often exponential. Exponentially, they are decreasing. So option B is also correct. We have to find out the wrong option. Okay. Then third is your stationary phase is the cryptic growth phase. Okay. So firstly, we should know the cryptic growth phase. Before moving towards that, let's check the option D. The rate of generation of new cell during exponential growth phase is constant. Okay, this is your exponential and the rate of generation. They are not asking about number. Okay, see the word rate of generation. They're not asking about number. So obviously, rate is constant in your exponential phase. Hence, the D is also correct. So wrong statement will be option C. But we should know what is cryptic growth. So cryptic growth means hidden growth of some individual. This means hidden growth of some individuals. Of some individual cell in starving bacteria in death cells. Means this is hidden growth, like you cannot see this growth distinctly. This is the hidden growth of some individual cell in starving bacteria that are there for the starvation phase. So in those bacteria, some individual cells are there which have the hidden growth, and in the bacteria in death phase. Okay, this happens in death phase. So stationary phase is the cryptic growth phase. No, it is your death phase is the cryptic growth phase. So instead of stationary here, if there was the death phase, then it would be correct. But the question is about the wrong statement. So option C is the correct answer. When we go for the this question, that is match the test in group 1 with its application. So for this, basically in the syllabus, it is not explained about this step, but we have to have a look on it. So we will see it later on. Okay. The next question is, which one of the following is not correct about the antibiotic resistance mechanism in microbes? So we all know what is antibiotic resistance mechanism, like how your bacteria is going to resist the dose of antibiotic. Okay. Now the not correct. That is, you have to go for the wrong statement. Options are mycoplasma is naturally resistant to penicillin due to presence of R plasmid. Okay. Next is gram-negative bacteria are impermeable to penicillin G. Okay, this is correct. Now the third is beta-lactam of bacteria can cleave penicillin. Yes, because in penicillin they have the beta-lactam structure. Beta-lactam ring is there in the structure of penicillin. Selective microbes can efflux penicillin entering the cell and develop the resistance. Okay, this is also true. Now not correct one is mycoplasma. This is wrong. Mycoplasma is naturally resistant to penicillin due to R plasmid. Why it is wrong? Because all mycoplasma, basically your mycoplasma lack cell wall. Okay. Mycoplasma lacks cell wall and therefore they are inherently resistant to beta-lactam antibiotics. To beta-lactam antibiotic, and this beta-lactam antibiotic are nothing but the example is penicillin. Why penicillin? Because penicillin has the beta-lactam ring in its structure. So the not correct option is mycoplasma. That is option number 8. The next question is, a suspension of photosynthetic green algae, it was eliminated in the presence of CO2 for few seconds. The first metabolite in the Kelvin cycle to be radio-labeled filtering. For this, we should know what is the Kelvin cycle. Kelvin cycle basically cycle that is going to take place in the three basic phase, that is carboxylation phase. This is the first one. Second is reduction. Third is your regeneration. Now the question is, what is the first metabolite? Now from when we go from the carboxylation phase to reduction phase, the first metabolite which is formed, that is ribulose 1,5 bisphosphate. Now let's see option ribulose 1,5 bisphosphate. So this is the correct option, ribulose 1,5 bisphosphate. Because in Kelvin cycle, it outlines the event that results in the formation of the organic molecule from inorganic. Like from CO2, you will get an organic molecule. So this whole process is outlined for your Kelvin cycle. Now ribulose 5-phos ribulose bisphosphate, it is carboxylated by CO2 to form a hexose bisphosphate compound. Means here you can see your ribulose 1,5 bisphosphate. From that ribulose 1,5 bisphosphate carboxylate is the compound, and then we get the 3-phosphoglycerate. Then from 3-phosphoglycerate we get 1,3 bisphosphoglycerate. Then from that glycerol there 3-phosphate, and then in regeneration it is again regenerated to ribulose 6-phosphate, and then further it goes to 1,5 bisphosphate. And here you can check the how ATP are consumed, and ATP is converted to ADP. How NADPH plus H plus is also converted to NADP. So here, like energy is being utilized for the reaction, and the first metabolite is ribulose 1,5 bisphosphate. So the correct option is option D.